Multiplex PCR, as its name indicates, here we can target more than one genes in the same reaction mixture. हमारे पास जीनोमिक डीएनए है किसी ख़ास ऑर्गेनिज्म का, या इवन जीनोमिक डीएनए अगर मिक्स भी है, तो उस सिचुएशन में हम एक ही रिएक्शन मिक्सचर के अंदर या एक ही रिएक्शन ट्यूब के अंदर एक से ज़्यादा जीन्स को टारगेट कर सकते हैं. अगर हम एक से ज़्यादा डीएनए सेगमेंट को या जीन्स को टारगेट कर रहे हैं, तो उसके लिए हमारे पास प्राइमर भी होने चाहिए. So we can say that we are going to use more than one primer so that we can target more than one gene or carry out their amplification within the single reaction mixture. So this process are the variation of basic PCR, where we are going to use more than one primer to target more than one gene in the same reaction. It is called as the multiplex PCR. This multiplex PCR, it can be used for different applications, as in case of the diagnosis of different diseases. It can be used. It can be used to find out the mutations in intron or exonic regions of the DNA, and as well as gene dosage effect can also be determined by using multiplex PCR. Suppose we are working on certain diseases, like diseases that may be caused by streptococci. Streptococci जो है, ये एक ग्रुप है माइक्रोब्स का जो कि राउंड होते हैं और ग्राम पॉजिटिव होते हैं. So they are commonly called as streptococci, and they may include different type of streptococci organism. Like they may be streptococci, lactococci, Vangococci, and some other type of gram pastococci may be there, and they may cause diseases like streptococcosis. That may be the disease of fish or other living organism. And now we had the idea that these organisms, they may be the responsible or the cause of this particular streptococcus disease, which is called as streptococcosis. And now how we can diagnose it? We have a sample tissue. We can even grow the microbes in the laboratory. We can extract the DNA, or even from the sample we can get the DNA. And this DNA we can use to amplify different genes. And those genes, they may be specific for particular streptococci organism. Like we can target the 16S ribosomal RNA gene that is commonly used to identify the microbes. In addition to that, other genes like 23S for another microbe, 23S ribosomal RNA gene, it can be used. For other genes, other determinants, either surface determinant of the microbes or other pathogenic determinants that may be related to certain toxins, they can be the target. Their primers can be developed. So, suppose we have targeted gene A, B, C, D, E that may represent a particular gene. We have the primer. Now we can use the same reaction mixture and can amplify different genes corresponding to maybe different region of the same organism's DNA, or maybe the DNA from another organism in a reaction mixture. It can be amplified, and then the results can be observed on by using gel electrophoresis device. We can separate the DNA fragments and compare it with the standard fragments. So in this way, we can have the idea that this particular disease, it is caused by this group of microbes. So in this way, by using single reaction mixture, we can amplify different genes. And it can also be used for the diagnosis of some diseases within the human, like the DMD disease in young males, which is called as Duchenne muscular dystrophy. Duchenne muscular dystrophy or DMD. So actually, it is the disease in the young males. It may suffer around this. It may be the case like 1 in 3600 individuals are young males. One may be infected or having this problem of muscular dystrophy, which is called as DMD. And actually, it is because of the mutation in dystrophin gene. So dystrophin, it is a muscular protein, and mutations in certain exons, it can result in the muscle degeneration, and it may cause premature death of the young males. And although this disease, it is rare in female, but it is caused, or it is mainly linked, or you can say that X-linked trait that cause this muscular dystrophy. Now, the mutations in different exons within this gene, it may be the responsible for this DMD disease. That is evident from this slide that we have different samples like 1, 2, 3, 4, 5 up to 10, and we have target different exons like exon 45 to exon 8. We have targeted, and this may be the one sample from an individual. This is from another individual, and here in the lane 7 we can note that here the middle band it is absent that corresponded to the exon 48 here. Exon 48, that is the main responsible here, and it is absent here, and this is because of the mutation, and this resulted in the Duchenne muscular dystrophy. And here I had quoted the reference of the worker who worked on this disease. So we can say that by using multiplex PCR, we can diagnose different type of diseases, either related to humans or some diseases that are related to pathogens like gram-positive streptococci or other microbes.